What I'd like to talk about today is the cell cycle. So a cell cycle could be divided into these two phases, interphase and the mitotic phase. Inter, like in between, like intermolecular hydrogen bonds. Interphase is the phase in between mitotic phases. And this picture, interphase is shown in green. And we can notice that interphase has some subdivisions. First subdivision, G1. This is a cell that has just formed from a previous cell division event. This cell needs to maybe increase its amount of cytoplasm, maybe needs to repair or build the plasma membrane, maybe needs to replicate some organelles like mitochondria. So during the G1 phase, that cell is growing. It's becoming the type of cell it's destined to be. It's going to be an onion root tip cell, or it's going to be a fish egg and grow into an embryo. Next, that cell enters into the S phase. I think of this as the go-to-work phase. When a cell is in its S phase, maybe it's doing its job. It's a leaf cell, part of a cell of leaf, and it's contains chloroplasts, and it's going to start to undergo photosynthesis. Or maybe it's a cell that lines the integumentary system, and it is going to start to produce mucus or keratin. So whatever that S phase is, that's the cell going to work. Some cells will get a signal from their DNA that they're getting ready to divide. So cells that are then going to divide need to double their DNA in a process we call DNA synthesis, or some books call it DNA replication. Not all cells divide. Think of your body, think of your neurons in your brain. Your neurons go through their own G1 when they're forming during embryogenesis and young childhood. And then those neurons formed your brain, and then forever, for the rest of your and their life, they will send signals across and send those action potentials. Your neurons will never go into mitosis. Your skin cells are quite the opposite. So when your skin cells are formed, they may work for a couple of days to weeks, producing keratin, covering or performing that first line of defense. And then also they get the signal to divide, and it's time for those cells to make a copy. They'll go through the DNA replication process, and then G2. G2 is when the cell prepares to divide. If it's an animal cell, the centriole pairs double. Sometimes it again increases in its cell size to prepare for cell division, etc. All of this is the phase in between cells, or interphase. If we want to take a look at a cell in interphase, we can see the edge of the nucleus. That's one of the key characteristics of a cell in interphase when we look through the microscope. All four of these plant cells, even though they've been stained pinkish purple, all of these are in interphase. I can see the edge of the nucleus, but I cannot see the DNA. In this cartoon, this is an animal cell. We have centriole pairs that are doubling. It's going to cause a region called the centrosome. But what I'm looking for is this. I can, yes, see the edge of the nucleus, but I cannot, it's difficult to make out the individual chromosomes. We see sort of a darker area, but I can't make out individual chromosomes, and that's how I know the cell is in interphase. By the way, in these plant cells, this item, that's a nucleolus. Remember, the nucleoli are where the ribosome subunits are made. And in this plant cell, we can see them. We can just barely make out a nucleolus in the background of this sketch. All right, so we have growth for G1. The cell goes to work in the S phase. If the cell is going to divide, that's when the DNA replicates. And if the cell is going to divide, that's where the rest of the DNA replication is completed, and the cell gets ready to go through mitosis. All right, so now I'm back to this yellow phase. In this pie chart, it looks like the mitotic phase is a much more brief amount of time, just according to this particular pie chart. Mitotic phase has the classic stages I'm thinking you've heard before: prophase, metaphase, anaphase, telophase. When I think of mitosis, I want to think, where are the chromosomes and what are they doing? Mitosis is all about what are the chromosomes doing and what are they going to do next? Cytokinesis literally means movement of the cytoplasm. Cyto, like cytoplasm. Kinesis, like kinesthetics or kinesiology, that means movement or study of movement. Cytokinesis means the cytoplasm moves. So the cytoplasm has to move to separate this now doubled structure into two separate cells, and we'll document that on the board in just a little bit. According to this plan, cytokinesis can happen after mitosis, or anytime during telophase, or maybe even at the very tail end of anaphase. Maybe this is a cancer cell and it's increased its mitotic rate, growing into a tumor. Hurry up! Cytokinesis can occur during the end of anaphase, throughout telophase, after telophase, or maybe not at all. And so it depends on the cell. So again, we're going to go through interphase. And when we identify it under the microscope, so we can see the edge of the nucleus, but we can't really see the DNA. So now I want to talk to you just a little bit about this DNA replication process. If a cell is going to divide, it has to double its cellular recipe. It's got to double up the recipe book with all those genes that make the proteins. That's DNA. Remember, the genes hold the directions for the synthesis of proteins, and basically it's the blueprint or the instruction manual for that cell. If one cell is going to divide into two, the cell has to double up its DNA. And so we see a couple phrases here, and what I'm going to do is I'm going to go on the board and I'm going to try and sketch as best I can my representation of DNA replication. You might look at this picture and remember the sugar phosphate backbone, ATCG for those nucleotide pairs, intramolecular hydrogen bonds, all that good stuff. So let's just take a quick view of how I'm going to represent this DNA replication. So what I'm going to do is I'm going to take this DNA. So here's my DNA. There's DNA replication. Hopefully you can read that. Just kind of, sort of. DNA replication during interphase. So I'm going to take my DNA strand, I'm drawing it as a line because I'm really good at that. You know the DNA is not a line. You know it's a double helix, ATCG, swirled into that helical pattern. But I'm going to zoom into that supercoiled structure wrapped around those histones. I'm just going to draw it as a stick here. And at this point, before it's doubled, I can call it chromatin. When I look into a cell under the microscope in interphase, the DNA is unwound, it's relaxed, and I call it chromatin. If the DNA is going to get ready to divide or double because that cell is going to divide, that DNA has to start to coil up and it's going to start to double. But I'm going to just jump to the chase and say, there it is. Now I have two identical strands of DNA. And these are called identical, or they're called sister chromatids. So we're changing the name a little bit, and that's okay. I'm going from this long, loose, uncoiled, not very easily visible structure during interphase of the DNA strands called chromatin. And now we're doubling it up into two identical sister chromatids. If you want to learn about this process of DNA replication, you should take GenBio 1. You can go through the whole process of matching complementary bases. You'll learn about the Okazaki fragments and the 3 to 5, 5 to 3 prime direction of how two identical copies of this DNA strand are made. But I don't need to do that. In this case, you're going to consider it's like someone handed you a textbook and then you made an exact copy of that textbook. So now you're standing there with two complete textbooks. Two identical sister chromatids. The next thing that happens is that these two identical sister chromatids sort of become attracted towards each other in a particular region. It's not always exactly in the middle. You take molecular genetics, you'll learn a lot more about this, and you should. But now they're going to become attached at a region I'm going to call a centromere. And we start to see, depending on your level of biological inquiry, couple different names for this region, but I'm just going to go sort of the non-majors bio realm and call this the centromere. So now what I've got are two sister chromatids attached at a centromere. It's like you took these two identical textbooks and stuck them together with a piece of gum for some reason. Now what's much faster is if I just draw this structure as an X. It's so much faster. So an X is two identical sister chromatids attached at a centromere, and now I call it a chromosome. And chromosomes are really easy to see in a slide. And chromosomes are going to be visible under the microscope. And I have my little molecular model kit here that I talked about in the mitosis lab. But these little dark stained structures on this plastic model, these are supposed to be the chromosomes. So chromosomes are visible. But chromosomes are like carrying around 2 copies of the textbook stuck with a piece of gum. Nobody needs 2 copies of the piece of textbook. 2 copies of a textbook. Maybe you need the piece of gum. But anyway, so here's a chromosome. And this is what we're going to follow through as we go through mitosis. So what I've done here is I tried to take us through this picture. I've tried to take us through the fact that here's my initial strand of DNA. It doubles up during this DNA replication process. And if you want to learn about this, you should. Look, a new nucleotide T is going to match into the existing nucleotide A. It's fantastic. And now we got these 2 identical sister chromatids, and eventually this structure sort of resembles an X. And that's our chromosome attached at the centromere. All right, if DNA is unwound and relaxed, we call it chromatin. What I tried to put on the board is kind of like this end of it. This is if we sort of stepped back and we just see that linear structure. If we zoom in, we realize how supercoiled that DNA is, wrapped around these histones, and eventually we see this double helix. Anyway, when we see this chromosome structure, we know that that's a cell that has to be getting ready for mitosis because it's got 2 copies of its genetic material. Remember this for when we not only go through mitosis, but next week when we talk about meiosis, or in a future lecture when we talk about meiosis. All right, so I've gone through interphase and I'm going to go through the mitotic phase. And mitosis, and I'll let you copy these down as I clean off my board. So mitosis is where the DNA, or where the chromosomes are located and what they're doing. So remember, mitosis is the location and the destination of the chromosomes. What has to happen in mitosis is the nucleus is going to dissolve and separate. Chromosomes, because they've doubled during DNA replication, they're going to have 1 set of chromosomes is going to end up in 2 future daughter cells, we call them. And cytokinesis is movement of the cytoplasm. Now these are phrases I took out of the textbook. So my job as the enzyme to your learning is to try to regulate or speed up your understanding of what these words mean. So let's do that. For mitosis, in this classic sketch that I figured out how to do on the computer, and I'm so proud, I think of mitosis as having 3 different purposes. One reason that cells might go through cell division is because they're growing, like a fish embryo. Another reason might be for repair, because you scratched your skin and you want to rebuild your integument. Another one is just to maintain tissues. And so cells like fat or connective or your blood supply, we have stem cells in our red marrow, red bone marrow that are constantly cranking out millions of new red blood cells. So just to maintain a certain level of tissue cellularity. We might just have this constant, monotonous, undergoing level of mitosis. When mitosis happens too much, too fast, without regulation, then we can consider that to be perhaps cancer. In these terms, mother and daughter, you don't have to use these words. You can say initial cell and resulting cell. You can say father, son, any combination. It doesn't really matter. But you realize that there's an initial cell here. It's going to double up its DNA. And then one set of chromosomes, a complete set of chromosomes, are going to end up in this resulting cell. And the other complete set of chromosomes ends up over here. So the mother cell with the two identical textbooks stuck together with a piece of gum, one textbook goes to this cell, the other complete textbook goes to that cell. And this initial cell doesn't exist anymore. Pro, meta, ana, telo. Pro, proceeds, that comes first in the procedure. Meta means in the middle, like metadata, metaphysics. Ana means to move. When blood vessels grow into a tumor, because a lot of times tumor cells give off chemical signals and they trick red blood cells to grow into them. We call that anastomosis. Ana means to move. And telo means the end, like a telephone or a telescope. So we have the first one, the middle one, the move one, and the end. Prophase, I'm going to let you write this down. I'm going to go back to the board and sketch my prophase and see if this works. In some scientific communities, prophase can be divided into several substages. I don't want to do that. Some books will have early, middle, late prophase or pro-meta. I don't want to do that. For this level class, I just want to say prophase. First thing that has to happen, we've got to get rid of the nucleus. Because the nucleus is holding on to the chromosomes. They've already doubled up. They're little X's. But we got to get rid of the nucleus so we can let the chromosomes out. Enzymes will do this. Then this chromatin, this long, loose, difficult to see, non-visible chromatin starts to coil and supercoil. And just as a quick flashback, that's one of the reasons why I show this slide, because I want you to realize how supercoiled this molecule is. So then during prophase, the chromosome become visible. So this chromatin, we've doubled up, we have those two pairs attached at a centromere, and then it continues to coil and becomes super coiled, and then we can see it. All of a sudden it pops out into the nucleoplasm and we can see it. If it's an animal cell, central pairs move to the opposite pole. This slide is an animal cell. I cannot see the centrioles, but I know they're in here. And I also know central pairs are over here. If we ran a different microscope lab and stained for the proteins in the centrioles, those little microtubules, then we could see the centriole pairs, but I'm not going through all that, so know that they're there. The central pairs have pulled between them more protein fibers, more microtubules, and they've built sort of a jungle gym, except your book calls it a spindle apparatus. And the spindle apparatus spans from pole to pole. It's like a tightrope walker. We have to give something for the chromosomes to hold on to. Instead of one tightrope, there's hundreds. So now we have all these little spindles that are going to span from pole to pole for the chromosomes to hold on to. The chromosomes then click onto that tightrope. They click onto the spindle apparatus at that region that we call the centromere. If you wanted to study this a little bit, get a little bit more technical, you might call that region the kinetochore, but I'm just going to call it the centromere. So I'll go ahead and I'll try to sketch my version of these cells in their various phases. And I'm just going to draw an animal cell. If I drew a plant cell as well, I'd just put a little box around it for a cell wall. But for a cell and interphase, I can see the nuclear membrane. For interphase, I can see the edge of the nucleus, but I cannot see the DNA inside. So I'll say the DNA is unwound, it's not visible, and at this point it's called chromatin. I don't know if we're in G1, S, or G2. And for lab, I don't care. You're not going to be able to tell me if this is G1, S, or G2. You'll just know that it's interphase because you can see the nucleus, but you cannot see the DNA because it's doing a DNA replication and it's unwound. But now I'm going to go into prophase. And so what happens now I'm in the mitotic phase. What has to happen in prophase, first of all, we've got to get rid of the nucleus. So enzymes come in and break apart the nuclear envelope. Nucleus is here, keeping the DNA. Nope, nucleus has got to go. The other thing, I'm going to draw this little barrel here, and these are my centrioles. And when you go into your textbook, and when you see the next slide, you're going to find a better picture of centrioles. Remember, they're those little microtubules, non-membranous organelles. But what's happened is the centriole pairs have doubled up during the interphase of an animal cell. And then the central pairs have moved to the opposite poles. They get pushed along these cytoskeletal components. And what the centrioles have been created, and I'm just going to span across, this is now the spindle apparatus. It's this three-dimensional, all these strands of microproteins, these little microtubules, the spindle apparatus is formed. The next thing that happened is since my nucleus was chopped apart, the chromosomes, actually, they're visible. They look like little X's now. Chromosomes attach. They click on to wherever they bump into a spindle apparatus. And what I'm going to do in this picture is I'm only putting 3 chromosomes. If we were to talk about mitosis, say in your skin cell that you're scraped out for the microscope lab, every cell in your body, except an egg or a sperm, all of our somatic cells have 23 pairs of chromosomes. That means 46 chromosomes. I do not want to put 46 chromosomes on this picture. It will be even worse that it's already shutting up the bit. So I'm just putting randomly 3 because that's what I feel comfortable with. You can put 2, 4, 6. You can put all 23 pairs of chromosomes if you want for an animal cell. All right, so that's my prophase. Next one, metaphase. What I love about metaphase, it's so easy to draw. So these are my centriole pairs because I'm making an animal cell. A plant cell doesn't need centrioles because a plant cell can attach its spindle apparatus right onto the cell wall. It's strong enough to hold it. But an animal cell isn't. But here's metaphase, and there's those centrioles, and there's my spindle apparatus. And the nucleus at this point is gone, it's just a memory. So what happens is the chromosomes kind of get pushed and pulled to the opposite pole. I mean, push and pull rather, to the center of that cell. It's almost like we've set up an imaginary equator. So now the chromosomes are pushed and pulled, and now they line up right at the center of the cell. So we'll go over and take a look at what's happening with our PowerPoint and see if we can identify these stages just as well. So here we go, prophase. The nucleus is still here, but it's dissolved already on this side. So prophase is kind of a crazy mess. Centrioles are here, the chromosomes are bumping into the spindle apparatus, and they're eventually going to attach. Remember, that's what the centrioles look like, not on my sketch, but the centrioles are these little types of microtubules as part of the cytoskeleton. We see this in animal cells, but not plant. And your Pearson textbook, just like what I have on the board, or I tried to, interphase, nucleus, yes, DNA, can't see it. It's called chromatin. It's long, loose. To me, it looks like a bowl of cooked wet spaghetti. Can't really tell where one starts and the other one stops. I don't know that this is G2, except that they told us interphase. Both of these are prophase. So what I tried to put on the board for prophase, I wrapped it into one picture. This has broken it down into 2, but we're just going to go with prophase. So let's get rid of the nucleus. Let's start to tighten up, coil up the chromosomes. If it's an animal cell, the centriole pairs move to opposite poles and they build the spindle apparatus between them. Let's click the chromosomes onto the spindle, whether we call this area the kinetochore, but it's where the centromere attaches. And now we've clicked on it. It's like the chromosomes are going rock climbing. They've just clicked onto their rope. Prophase. My favorite, metaphase. All the chromosomes lined up in the middle. I think this is a lovely depiction of the centriole pairs, the three-dimensional spindle apparatus. And here's my chromosomes. They've been kind of pushed and pulled, pushed and pulled. And notice how they're lying. It's not really exactly like my picture on here, but notice there's one textbook stuck with a piece of gum, and there's the other textbook. Because remember, these are two sister chromatids attached at the centromere. That's metaphase, and it's one of the easiest phases to find under the microscope. Next one, anaphase. So we take these X chromosomes and now we break them apart. We take our two textbooks stuck with gum and we separate them and we clean the gum off. And now we have two textbooks. And one textbook is going to head along the spindle apparatus towards a pole. And the other textbook heads along the other spindle apparatus towards the opposite pole. Ana means to move. Now, hopefully you've had enough time to write these down. The sister chromatids are split by the centromere. Now we can call these daughter chromosomes. They're complete, fully fledged daughter chromosomes. And so what I've started on my little sketch here is I've tried to create my centriole pairs are still in the opposite poles. And one of these chromosomes, or each one, splits. And this chromosome is now heading with its centromere along the spindle apparatus towards the opposite pole. Think of it like a Pac-Man game as it's digesting that spindle apparatus. And the other daughter chromosome is now attached by its portion of the centromere. Ching, 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 ching. And they're actually moving to the opposite poles. So because I put 3 chromosomes on here, I'm just going to complete my story and think of those 3 chromosomes as moving to the opposite poles. What happens in anaphase, though, is we actually still have some remaining spindle apparatus proteins. And in a really fun twist of events, which I'm not going to test you on, but it's kind of cool, each of these chromosomes, as they move, actually attaches the microtubules that they've cut off onto remaining microtubules, which pushes the poles apart. So as these chromosomes digest the spindle apparatus, they also actually cause elongation of the remaining microtubules. Go figure. Ana, to move. And now we want the end. Telophase. And I'm going to make my telophase pretty close to the end. Try and draw an hourglass. So here are my centriole pairs that looked much better on the picture because this is an animal cell. I could run this whole process again for a plant cell, but I would change it by putting a cell wall on the outside and omitting the centrioles. And I would put my spindle apparatus right onto the cell wall. All right, so here's telophase. My chromosomes have now reached the opposite pole. They arrived. Hi, it's for dinner. And then little enzymes come in and capture these newly arrived chromosomes and trap them in a nucleus. So a new nuclear envelope forms. So these 3 chromosomes, after a long day's journey, have just reached the pole and then they're surrounded by phospholipids in a bilayer. Enzymes come in and build a new nucleus. And the same thing happens to these chromosomes that arrive down here. They get caught or trapped into a new nucleus because we need to then hold the genetic material in a nucleus because this is a eukaryotic cell. Bacteria don't do this, not like this anyway. All right, in the meantime, the spindle apparatus has been getting longer and longer and longer, pushing those poles apart. And eventually, this area kind of gets skinnier and skinnier. And eventually what happens is, bloop, and it separates or breaks completely. And so, after these two cells separate completely and make the bloop sound, now we have two separate cells. And this process I called cytokinesis. So cytokinesis already happened. And this little molecular model kit that I have, this is trying to show chromosomes that have reached the opposite poles. A new nucleus is going to form around each pole. And meanwhile, we start to see the cytokinesis. As the poles are pushed farther and farther apart, eventually, just like if you had a big soap bubble in the kitchen sink, and if you could hold on to it, try to do this, it's very tricky. Break the soap bubble, you get two soap bubbles. That's what happens here, cytokinesis. So I'm back to my anaphase, and here's showing a molecular model kit, just like the one we have. Love the kit. In an anaphase, you could have the chromosomes have moved a little, they've moved a lot. These are the centrioles and the poles. But the point is, anaphase, the chromosomes move, still have a little movement to go. In telophase, the chromosomes have reached the opposite poles. They can't go any farther. That's it. They've gone as far as they can go. This is a plant cell, and I know it's a plant cell because of the square shape of the cell wall. These are the chromosomes. There's one complete set. Here's another complete set. We still see a little bit of the remnants of the spindle apparatus. And then what's going to happen is the plasma membrane of this plant cell will separate, but then the plant cell will build a new cell wall between them. We have two separate plant cells. In an animal cell, here are the chromosomes. They've made it to the opposite poles. They're going to start to unwind. They're going to kick back and relax back into chromatin. Just give them a couple minutes to hours, and we're not even going to see those chromosomes again. They're just going to unwind and sort of disappear into their nucleus. And now we see cytokinesis. Some people call this the cleavage furrow. This is the furrowing. Here's that separation of the two, and eventually, bloop, this will break, and we're going to end up with two separate cells. So we take it through meta, ana, and then in telophase, we see this invisible enzyme workers have come in and they're rebuilding a new nucleus for these chromosomes. Enzyme workers over here are building a new nucleus for these chromosomes. Meanwhile, cytokinesis is underway. And here's our cleavage furrow in an animal cell with a pinch and then the break. In a plant cell, I mentioned that a new cell wall is built. So we have Golgi apparatus and other cells, other organelles rather, start to make little vesicles of the polysaccharide cellulose, because that's what plant cell walls are made of. So it's like we get little wheelbarrows full of cellulose dumped in between these two cells. Eventually they form together. It's called a phragmoplast, but I'm not going to ask you that. But we start to see a new cell wall. Now you get two separate functional plant cells. So the take-home message for mitosis, we're starting with an initial cell, call it mother, daughter, parent, doesn't matter. We get two genetically identical offspring cells. Daughter, son, offspring resulting, it doesn't matter. But these two daughter cells are the exact same as each other. They have the same DNA, the same number of chromosomes. They're the same cell. And so we get pro-meta-anatelophase. We get it one time through. We get two cells and they're identical. This all seems very logical, and it is, but we want to remember this and be comfortable with mitosis, because in a couple lectures from now, I'm going to cover meiosis, and that's the production of gametes, and it doesn't run exactly like this. It's similar, but it's not the same. So it's good to learn mitosis first because it's sort of the easy, logical one. Meiosis is easy too. We just have to work a little bit harder to get through all the divisions. All right, if you want to find a YouTube channel, there are many, 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 many online sources. I have posted some. There's some within your lab manual. If you can find a video of watching mitosis running its way through, that's probably one of the best ways to learn it. This is one, and when you click on this as you watch it, go ahead and investigate the cells. These are what your animal cells are going to look like in lab, exactly. These are what your plant cells are going to look like in lab, exactly. And so after we've done the animal and plant lab, we can go back and identify those couple. Cytokinesis. And so that brings us to the end of mitosis. Excellent.